before, the proteome comparison tool in PATRIC is the second in our series of genomic comparison tools. And it's a bidirectional blast analysis tool where you can do a detailed analysis of up to 9 genomes compared to a reference. The tool in PATRIC relies on BLAST. I'm sure that almost everybody in the audience has used BLAST at some point. BLAST stands for the Basic Local Alignment Search Tool. And I'm sure you all know it allows you to compare nucleotide or protein sequences to each other or to a database, and it will calculate the statistical significance of the matches that it's found for you. BLAST can also be used to find regions of local similarity between sequences, and it can be used to infer functional and evolutionary relationships, but not only between sequences, but it can help you identify members of gene families. It can be useful in identifying homologs, orthologs, or potential xenologs. I've always known homologs and orthologs, but apparently xenologs is a new term that has just been, that I was just recently introduced to, and that's identifying something that's come by lateral transfer. The proteome comparison tool in PATRIC uses BLAST-P, which is a protein-to-protein blast. I'm going to keep making this point because often we get people who are trying to submit nucleotide sequences. The tool works with the proteins and not with the nucleotides. So why should you use BLAST to compare genomes? Well, generally, if you have a new sequence, BLAST is one of the first ways when you're searching for similarity that gives you a lot of information about your sequence or your genome, and in any new genome, that's one of the first things you start thinking about. Bidirectional BLAST hits, which is what the proteome comparison tool uses, that is commonly used as a proxy for defining orthology in comparative genomics. So looking at gene families and trying to decide which genes belong together. The best bidirectional hit is found when the proteins encoded by the 2 genes, each in different genomes, find each other as the best scoring match in the other genome. And I have a cartoon of this in a little bit to show you what I mean by that, because sometimes people are a little bit confused about bi and unidirectional BLAST hits. Now, I've mentioned orthologs. People who do comparative genomics talk about orthologs all the time, but perhaps it isn't a term that some of you are not familiar with. Orthologs are defined as genes in different species that evolved from a common ancestor or an ancestral gene. Now, what's important about orthologs, and the dogma is that orthologs retain the same function in the course of evolution. And so in order to predict function in a new genome, you identify the orthologs, and generally that's seen as a reliable prediction of gene function in newly sequenced genomes. So let's talk a little bit more about PATRIC's proteome comparison tool. You can use it with bacteria or archaea, and it supports the proteins in the genome. So we're looking at the comparison between the protein sequences. It can be used for genome-wide comparisons, or you can use it looking at a certain set of proteins that you're interested in and can see how broadly that is shared across a number of genomes. It gives you bi and unidirectional BLAST-P hits. It enables the comparison of proteomes from whole genomes or sequences, which I mentioned before. You can create a group of, let's say, type 3 secretion systems and look across different species within a specific taxon and see how broadly it's shared or if there are any problems with those genes. Now, there are several tools that do this kind of stuff. The PATRIC Proteome Comparison Tool is different in a couple of ways. One is that it's an online tool. It's not a standalone command line program. You can launch this via the website, and it will return your results for you there. You don't have to know any programming to be able to do it. Something else it does is it produces an interactive and publication quality image that you can use directly in a publication, and I'll show you some examples of that today. The thing I like best about the tool, of course I like that it's online because I'm a biologist, not a computer scientist, so I'm glad I don't have to do it in the command line, and I like the figure that it produces, but it produces the most valuable thing to me is the downloadable text file that shows you each of the genes and the blast hit parameters assigned to each of them. So it's showing you download the text, and it's showing you exactly what the figure shows you, all the details behind that. Now, it will show you homology and unique regions in the reference genome. It is a bidirectional blast analysis, but it's based on the reference genome that you choose. So I'll caution this to you a couple of times today. If you've got genome A as your reference and you're looking at genome B, it's going to show you all the genes in genome A and how they compare to genome B. If there's a large section in genome A that's missing in genome B, you're going to see that. If there's a large section of genome B that's missing in genome A, you won't see that. So you need to relaunch the tool with genome B as your reference genome in order to be able to see the complete pan-genome of the two genomes that you're looking at. So this just describes basically this cartoon, a bidirectional or a unidirectional best hit. The bidirectional is pretty easy to explain. You can see that in the cartoon in the lowest boxes in the green genome and the blue genome. When they're blasted against each other, the best hit is the one that they're pointing at. So they each recognize each other as the best hit. A unidirectional hit is at the upper part of that cartoon. Genome, or I'll call it green genome, has 2 proteins, and blue genome has 1 protein. The uppermost protein recognized in the green genome has its best hit as the gene in the blue genome. But that gene in the blue genome recognizes the lower one, and the green one is the best genome. So that's a unidirectional hit. So I talked to you a little bit about the interactive image that we provide in PATRIC. Generally, you have the circular view. I'm mousing over it so you can see it here. And what the circular view includes is there's the list of genomes that you can see here that goes from the outside to the inside. And you'll notice that there are a bunch of different colors. We have a key to the color that shows you the best hits and the unidirectional so that you can sort of look through that and be able to see the differences. And it's also showing you the sequence identity. So I guess you could say blues, good, reds, not so good. So when you see things that are tightly, their sequences are very close to each other, the darker blue that is. As it moves away from the blue, it gets to things that have less sequence identity. Now, if you were to scroll in, which we'll talk about later today, and look at specific regions of this, the image is also interactive. I can click on an individual band in here, and it'll take me to the gene landing page in PATRIC. That's the place where we assemble all the data that we currently have for that gene. Let's say it's a tuberculosis H37RV genome. We have a lot of data for that organism. We bring in a lot of transcriptomic data. We try to bring in a lot of data for certain genomes in PATRIC. So if you were to click on that, it would take you to the gene landing page where you could see the genome browser. You could see the gene neighborhood. That's the compare region viewer for that gene. It would tell you if there was involved in any pathways, if we had any expression data associated with it, if we knew of any protein-protein interactions, any correlated genes. We also calculate for a certain number of genomes patterns of up or down expression, and also literature. You'll notice that in some of the genomes, there are bands, and they're missing in others. So it's an indication, this this figure also gives you an indication of areas where the comparison genomes are actually missing data. So that's the picture that you can download in PATRIC. But as I told you before, what I like is the downloadable text file, which gives you a detailed comparison of your reference genome and up to 9 comparison genomes that are compared to the reference. Now, the reference and the comparison genomes all share a number of things. We tell you the contig that the gene is on, the order that the gene is on that contig, the size of that gene and the amino acids. We provide the PATRIC and RefSeq locus tags because we understand that many biologists speak GenBank and RefSeq locus tags. That's how things are referred to in papers. We want to make it clear and easy for you to draw the correlations that you need between data types. We also give the gene name and the functional annotation. Now, the reference genome has something that the comparison genome lacks. And for the reference genome, we'll also show the start and the end location for the gene on the specified contig or chromosome. And we'll also indicate the strand that the gene occurs on. That's if we have a genome. If we just have a series of proteins that you've used, you won't get that sort of information. The comparison genome is where the blast hit details are stored. The very first line or column under in the comparison genome information will tell you if the hit between the gene and the comparison genome and the reference genome, if it's a bidirectional hit, a unidirectional hit, or if it's missing. Some other information that's also included in the comparison genome information is we provide the percent identity of the blast hit and the sequence coverage. Both of these are important. The percent identity just is basically telling you how close these genes are together. Are they 99% similar? But the sequence coverage is an important clue you need to look at because you could have a very small region. Let's say you had a gene that was pseudogenized and it was just a piece of it. You would have a very high percent identity, but a lot of the gene would be missing, so there'd be low sequence coverage. So those are some things that you need to pay attention to when you're looking at the file. Now, I told you that our images can be put into publications, and here's an example. I generally try to keep track of all the papers that cite PATRIC, and I try to look at them to see how they're using PATRIC, so I know very well when people are using our tools, and this came directly from our tool and into this publication. I know that several of you from Clemson are interested in metagenomics, and so I included this one just for you guys. This is a pretty interesting paper. They are using our tool to evaluate how good metagenome assembled microbial genomes are. And you can see that that's what they were doing there was taking their assemblies that they got from metagenomic samples, and then they would compare them against a close reference strain in PATRIC. You can see from the pictures that how strong the homology is between the metagenome and the reference there. But another thing that can be used in this, when you use this kind of service, it can give you an indication of if your metagenome is missing a large chunk of the genome. I've seen a couple of people using it this way. So it's not just for genomes. You can take metagenomes and look at those as well. So what kind of sequences can you compare in the proteome comparison tool? Any of the public genomes are annotated in PATRIC, any of your private genomes that you annotated in PATRIC, but you can also bring data that perhaps you didn't annotate in PATRIC that's been annotated outside of PATRIC. you can bring that in and look at that too. We aren't prejudiced against anybody's annotation system, and we want you to make the kind of comparisons that are important to you. Another thing you can do, and I do this quite a bit now, I'm liking it more and more. I'll create a FASTA file of proteins that are of interest to me. Let's say all the virulence genes in a certain genome, and I want to see if its close relatives also contain those proteins. So I can upload that FASTA file and use it as my reference genome, and then the bidirectional blast, then I can load in a number of comparison genomes, and I can see how strongly those comparison genomes contain that FASTA file. Let me point out yet again that the tool requires protein sequences. Occasionally people will come and they'll give me a FASTA sequence that's nucleotide. The tool won't work with that, so don't do that. So here's what we're going to do today. We're going to submit 3 proteome comparison jobs in PATRIC. In the process of submitting those, I'll help you look at how you locate private or public data or reference genomes and how you select a reference. We'll also look at uploading and using a FASTA file for the reference genome or for the comparison genome. And we'll also look at using a feature group as the reference. I'll show you how to monitor the progress of your proteome comparison job. Now, these jobs generally run very quickly. If you're using it with a feature group, it can be less than a minute. Well, let's say less than 2 minutes. I don't want to get too optimistic. Some of the other genomes, I've never had it take me more than 20 minutes. It all, though, depends on the queue and how many people are lined up ahead of you. I'll show you how to locate your proteome comparison job so that you can find it and look at the results, and then we'll examine the results together. So we're going to get started, and I had something totally different prepared, but then this morning I was looking at a paper on Mycobacterium leprae, and I just put together a few examples based on that genome. I mean, who doesn't love Mycobacterium leprae? It's the causative agent of leprosy. It's a really interesting organism because it's undergone extensive genomic reduction, and they've dropped, compared to something like tuberculosis, which has 3,000 or 4,000 genes. Mycobacterium leprae generally has about 1,000, and it's highly pseudogenized. It divides very slowly. It's generally only in humans or in armadillos, so we're going to look at that, and we'll make some comparisons. We can't do anything as awesome as this Nature paper, but we'll look at differences that we can make in Mycobacterium leprae. So that's ending the talk. Now we're going into how to launch the tool, how to find data in PATRIC and launch the tool. So I'm logged in here, and I'm on the home page. So let's say I want to look at Mycobacterium leprae. I've read this paper, and I want to look at some of this data in PATRIC. One of the ways you can find data and find out things that you want to compare in PATRIC would be to look at the Organisms tab. When PATRIC was originally started 14 years ago, NIAID was especially interested in these bacterial pathogens. They were worried about bioterrorism. They wanted us to concentrate and provide direct links to these organisms so that researchers who were looking at them, working with them, would have direct access. I point this out just to tell you that even though we have this under the Organisms tab, we have all bacterial genomes and all archaea genomes in PATRIC, and we also have some eukaryotic hosts. So if you're not interested in one of the top pathogens, you could use the global search up here at the upper right to search for an organism, or you could go to all bacteria and drill down. But we want to look at Mycobacterium leprae, the causative agent of leprosy. So I would click on the Mycobacterium link here, and that's going to take me to the landing page for Mycobacterium, the genus. And you can see that wow, PATRIC currently has, you can see up here in green, 12,262 genomes that are part within the genome, within the genus Mycobacterium. That's a lot of information. You can't pack it all into a single page. So you can see we have a number of tabs across the top where we try to filter down the information for you. And every page in PATRIC is similar in that way. But I want to get to leprae. I mean, there are several ways to get there. But let's click on the taxonomy tab. And then I'm going to scroll down. It's in alphabetical order, and there are a lot of species within this genus. And I just scroll down, and I'm trying not to scroll down too fast because it always makes me a little nauseated when people do that and I'm watching. And then I clicked on Mycobacterium leprae. Before you click on it, look at that vertical green bar. There's nothing there. If I click on one of the boxes, the check boxes in the first column, it gives me an indication of all the possible downstream steps I can take once I've made a selection. This one tells me I can go to the taxonomy. So I'm going to click on the taxonomy view. Okay, that opens up the data for this particular species, and there are 13 genomes. To find out information about those genomes, I'm going to click the Genomes tab. So here are the 13 genomes that we have for Mycobacterium leprae. There are some things I want to point out. I'm going to make it a little bit smaller here so you can see this. The number of sequences and the number of hosts that we have here. You can click on the column head to sort things alphabetically. So I can see that 1, 2, 3, 4, 5 of these genomes were isolated from the armadillo. Now, I don't know if you all know this, Mycobacterium leprae is extremely host specific. It's generally only found in human beings, but it has been identified that it will infect armadillos. And when it came, it's an old world disease. When it came to the new world, there was the 9-banded armadillo that exists in Central and North America that became an adaptable host. And I mean, I don't have time to go into it, but it's a really fascinating story about how they use these as the host animals, the experimental animals for this organism. Also, you can infect a mouse just in the pad of its foot. That's the only place that it'll get leprosy. So I was looking at this and thought, I wonder if I could find any differences in leprosy based on the host. So that I decided, let's just try that. Let's have some fun and try it. So I'm going to keep this tab open so that I can come back and forth so I can be sure to have the right host name. And then I've got a new tab opened with PATRIC in it. I'm going to go into the services, and I'm going to click on Proteome Comparison. It's here under the Protein Tools. So I click on that, and it opens up the box for the proteome comparison. And you notice that the submit box is gray. It has to be blue for me to actually submit a job. So I have to choose things, fill in things in each of these 3 boxes before I can successfully submit a job. So let's first talk about the parameters. One thing that you can do is you can click to open the info dialog box, and that'll tell you what the standard parameters that the tool is set at. If you feel particular about that, you can open it up by clicking on it, and you could change the coverage, the percent coverage, the percent identity, or the blast e-value. I generally am not that picky, but I welcome you to do whatever you like. You have to identify a folder that you want to store the data in. That's one of the requirements in PATRIC. I have a number of folders here. You see I've selected one already. So I can go in, and I'm just going to show you how to name a folder. I already have one named, but let me show you how to do that. So you click on the folder icon, and it opens up a page, and that's telling me how many folders I have in PATRIC. If I want to create a new folder, I'm going to click on this, and I would call it Proteome. Oh dear, everybody gets to see how poorly I spell. Proteome comparison. And you could create a folder of that if you wanted to. But I'm not going to create one because I already have one. But let me tell you that once you create one, you click the Create button, then you'd have to drill down to find where that folder was. This seems to be freezing up on me a bit. Let me re-engage that. And once it was there, you could pick on it that way. Select it, see, from the down tab. So I selected it. Then you have to pick a name for it. So what I'm going to do is I'm going to do Lepre TN, that's the strain of the species, reference, and then I'll just say host, so I know what I did. I use this tool a lot, and one of the things that I've learned is to be very judicious in writing the correct name, or I have to open each one to see what it was. So the parameters are filled. Now we have to select a genome, and you could click on the eye to see the information about the reference genome. You can select a genome from PATRIC, you can upload a FASTA file, or you can use a feature group. So here's a little, what's supposed to be a filter icon. If I click on that, you can see that I can drill down. I can look for reference genomes, representative genomes, public genomes, or my genomes. If you have private data in PATRIC, look, I'll just deselect off everything else. And then I can see what private data I have there. And the private data you can always tell because it's got this little padlock in front of it. But you know what? I'm just going to click all of these. And I want to show you how awesome this search is. So I can put in Lepre, and then look, it's already looking. It's trying to fulfill my every wish. And then the TN strain. I chose the TN strain because it is a reference strain. GenBank and RefSeq have identified certain genomes that they consider the gold standard genomes. They're reference and representative genomes. They're the genomes that a lot of researchers do analysis on, and they have a lot of data associated with them. So that's what I want to choose. So I've got my reference genome. I still can't submit. So now I want to do my select genomes, the comparison genomes to add to it. Now, I've learned just recently that it's very important the order that you add these things. The first genome that you add to this table will be the innermost circle. Your reference genome is the outermost circle, so you need to add things in the reverse order that you want them to line up. I know that Mycobacterium leprae, TN, is an armadillo genome. So I want to look for genomes that I want to add those last because I just want to have them be close together so I can look at them. So I'm going to go over to the table, and I want to add something different. So let's start with a mouse genome. So we'll add this AL5 genome. So I'm going to go back to the table, and let's see what happens if I type in here AL5. So look, right down there it says Mycobacterium leprae AL5. So I click that, but I have to get that genome into the selected genome table. See, I still can't submit anything. To do that, you have to press this plus icon. And there it is, and suddenly, look, if I wanted to, I could submit this job, but we want to add more than that. So another one was AL5. So I'm going to go over here, click AL6, sorry, I can't even, and look, right away it knows who I'm talking about, so it brings it up. So right now the two innermost circles will be these two Mycobacterium leprae genomes that were isolated from mice. Let's add in some humans here. So look, we have this, well, BR4923 is a very well-known genome, so I'm going to add that in. So I just start typing BR, and it knows right away. So it will auto-fill it for me, and I have to add it in. The next one is this 7935681. I don't want to remember that. So I go over here beyond the green bar. You notice when I have a genome selected, beyond the green bar, it will populate all the information about that genome that we have in PATRIC. It'll tell you links to different resources. It'll give you comments about this, where this genome came from, where it was isolated from. So I'm just going to put that number here. And oh, wait, there are two copies of the same genome. What should I do? Well, let's go back to the table. There are two copies, yes, of the same genome. They have two unique identifiers in the genome ID. And let's look at the differences between these. One of the genomes has 176 sequences. It's a whole genome shotgun, so it's not a complete genome. This one has 107. And I would normally say, let's choose 107 because that's a, well, generally that's the fewer contigs is generally an indication that something's a little bit better quality. But when I look at the number of genes here, this one actually looks like it's closer. So these are the kind of decisions you may have to make while you're looking at these things. So if I select this one, it's the 0.3. So I'm going to go after that one. And I'm going to put that in. And there it tells me which version of the genome. And I'm adding that. So now we have 2 mice, 2 humans, and let's add in the armadillos ones that'll be next to it. So here we have 8T70, 8T70, and I click on it, and I add it. The next one is 7Q90. And I'm hoping you're seeing the power of the search thing. Sometimes I can just have the strain name without adding in all the information about the genome. And let's just add one of these because 5L116 has the fewest sequences. So I'll just add the genome ID for that one. And then I need to click it and add it. Now let me point out here that I don't need to just use leprae. I could add in I could add in tuberculosis. I could add in Escherichia coli. That's the beauty of this tool. What I'm showing you right here is because I was interested in leprosy. So I wanted to do a detailed genome comparison, but I could use this to search for homologs and orthologs at a much broader taxonomic comparison. So we have it ready, and we hit submit, and we're off. So once I hit submit, it tells me that this job should be finished shortly, and check the workspace for results. Down here in the lower right, you'll see that you can scroll over the jobs, and it should tell you which jobs have been submitted, which are running, and which are completed. Now, you were with me. We just launched this job. It said it's launched, but when I scroll over here, I don't see it under running or queued. It says 0. That's because sometimes, especially when the service is being used, there's a little bit of a delay in it being recorded in the jobs record, so don't be worried about that. Let me point out something else that will be helpful for you here too. At the top of each page, the eye icon gives you the information about the tool, but here is a tutorial. We'll have a direct link to the tutorial that tells you how to use the tool. I'm just talking to you today. There's no way you're going to remember everything I said, but if you click on these little, it's supposed to be a bunch of books. If you click on that, it'll open up a new window and show you exactly how to do the comparison in PATRIC and use the proteome comparison tool. In addition, I do some things that show you how to format the Excel file, and we'll try to get to that today before this is over, but all that information is here. Okay. So there was one other thing I wanted to show you. I'm going to go back to this page. So we have the proteome comparison. We'll look at the jobs in a minute. But let's go back to Mycobacterium, and we're going to the landing page because there's a specific organism I'm interested in, H37RV, the causative agent of tuberculosis, and I found that in the reference, in the list of reference or representative genomes. So I want to show you how else you could use the proteome comparison tool. For every genome in PATRIC, if we have specific databases that are for virulence factors, antibiotic resistance genes, human homologs, or essential genes, we have that information here under our specialty genes. And one of those is the essential genes. This is something that we do in PATRIC that looks at using a flux balance analysis in a computational pipeline. We try to call what genes the organism needs to survive. H37RV, Mycobacterium tuberculosis, is an intracellular pathogen, but it has a much larger genome than the leprosy genome. So I'm interested in, do these things share the same essential genes? To get to that, I could click on this number, that's 215, and that will take me to a table that lists all the essential genes in PATRIC for this genome. So I click up here and collect them all, and notice when I collect them all 215 of them, there are a number of things I could do once I've collected them, but I want to create a new group. So I'm going to select that, and existing group, I don't want that, I want a new group, and so I'm going to call it Essential Genes H37RV, and I'm going to add that group. Okay, and it says here, group added, Essential Genes H37RV. Look down at the jobs, by the way, you notice that my job is running when I look at one. Okay, let's go back. Let's open the tab for the proteome comparison tool again. And this time, we have to, once again, establish the output folder. I'm going to call this H37RV Essential Genes, leprae. Okay, so that one is done. Then I want to go into the feature groups. And look, the very one I just created is at the top of the list. So I click that, and that's going to be my new reference genome. It's going to be specifically against these genes. So let's just choose a couple, just for speed's sake. We're going to choose a couple of leprae genomes to compare to this. The TN, which is the reference, and the BR, which is BR4923, is a Brazilian isolate. And this is going to see if these two genomes, how strong the homology there is between them and the essential genes of H37RV. Moreover, we're going to see if any of them are missing. So we can submit, we have the blue light, and let's submit the job. So I've submitted 2 jobs in PATRIC. How do I find those? Double click on this jobs at the bottom page, and that will open the job. And you can see I've run a lot of these today, right? I've been very busy with running different jobs. And these aren't quite ready yet, so I'm just going to show you what a job I ran previously today looks like. So you select that, and once you select it, the green bar becomes populated with things you can do. I select the green view icon, and it takes me to this page that actually I don't care that much about. I don't look generally at any of these. The one thing I usually pay attention to is how long it took this job to run, which was 11 minutes and 37 seconds. But then I want to go here as quickly as possible, the blue eye at the top of the page. I realize we're having you click here, and now we're having you click here, but there are reasons for that. And it depends on, the reasons are based on which database we want you to look at. So remember how when we originally arranged this, we started from the outermost, all of these guys up to this AL6. These are all strains that were isolated from armadillos. And then the two inside are, this one is a mouse, and these are all human isolates. So we have this nice picture, but what does it mean? You could scroll in and look at things, but the real power in all of this is, to me, is in this file that we download the genome comparison table. When I download it in Firefox, and I'm going to save the file, I have to go into, well, that's not where I want to go. I want to get that. Oh, there it is. So I have to click on it, and then I want to open it with Excel. And then well, this is one I've already prepared, so we just have to wait for the other one to open. Maybe it opened behind this. Oh, well, here it is. I should have brought it up to the top. Okay. No, this is an older one. Well, we'll just talk about this one. I'm not sure why this one hasn't opened up yet. So for Mycobacterium leprae, this was my reference genome, and like I told you, it lists the contig, the order that the gene occurs on that contig or chromosome, the size here. So this is the contig, the order that the gene is on the chromosome, the size, the PATRIC locus tag, the ref-seq locus tag, the gene name, the functional annotation for that gene, and where it occurs, the start and stop sequence in the gene, and this tells you which strand it's on, the positive or the reverse strand. It tells you, then we get into the compare, tells you if it's uni or bidirectional, the locus tag, the order. This is important now because often you have to pay attention to these things. You notice here that these specific contigs are the 186 contigs, but over here it's on the 196 contigs. If you're making an inference that these genes are located next to each other, you're wrong. They're on different contigs. They don't necessarily belong to each other. So you need, although they could because it's a genome that's still in pieces, but you need to pay attention to that. This could also help you improve your assembly by looking at the data this way. So that tells you the gene order, the size, the locus tag, if it had a ref-seq locus tag, which it doesn't, if it had a gene name, which it doesn't, and the functional annotation of this gene. And then we get into these two that I have manually, functionally done conditional formatting on, which I'll show you here. So I select both columns, and I go into conditional formatting, and I say I want to highlight the skull rules, because what I want to do is rapidly scroll through all of these genomes that I've compared and look for differences. So less than, and let's say 0.9, and we'll color it green, less than 90% coverage or sequence homology. And then I've got to get rid of those blanks. So I highlight the cells that are equal to, and this is all in a tutorial too. I know it's a lot to scroll to tell you, but you can do this kind of thing pretty quickly with the tutorial. And then you can see patterns within your genome very quickly of things that have questionable coverage and things that are missing. So I wanted to show you this one that I worked on earlier. And what I did was I colored everything that's gray up at the top are the isolates that were from armadillos. The yellows are the isolates from humans, and the brown is from the mouse. And I also included H37RV. But I can use this to scroll rapidly through a genome and see areas of loss or gain. Like this one, this whole block of genes here in my reference seems to be missing across all of the other leprae genomes except this other one, which is a different. So those are the kind of things you can explore very quickly. So let's look, for example, this gene here. I just picked it out. I don't know much about it. That it has a 97% sequence identity, which you would think, that's really great. But then you see it's only got a 50% coverage. So paying attention to those kind of differences can be very important in helping you determine how good these genes, how good of a pair they are. So I know we're running out of time. I want to go in and look at this job I just submitted on the essential genes. So let's look at that. We click on it. The green bar says, view the job results. It also has this report an issue. If it failed for some reason, let us know. We'll look into it. We want to make this service extremely easy for you to use. Also, if you have suggestions, put that in. So I want to view it, and then I want to view the job. And so look at this. So in the outermost one is the H37RV, and then we're looking at the essential genes from that organism, and we're comparing it to two Mycobacterium leprae genomes. And you can see that the homology isn't so great. Some of it's like 80%, but look, a gene like this, it's totally missing. Do I need to open up the Excel sheet to see this? You can just click on that, and it'll show you that gene in PATRIC. So this is one that was totally missing in the leprae genomes. What is that gene? It's this gene that I'm not going to even try to pronounce because I'll ruin it. But you can see that it's involved in this pathway in PATRIC. You can also see where it's located within the genome. You could look at the genome browser. This particular feature that I'm going to click on is called the Compare Region Viewer. And that looks across the gene neighborhood, and it shows you what the gene neighborhood is like. It looks for a conserved neighborhood. And let me shrink it down so you can see it. This is just open for the reference genomes, but it's able to tell you how broadly shared and how conserved not just the gene is, but the neighborhood associated with that gene. It's a pretty cool feature that we have in PATRIC now. Also, if we have expression data for this particular gene, which we do have, we have a number of two-channel microarray samples that we've brought into PATRIC for a certain number of genomes, and H37RV is one of them. You can see which experiments it's significantly expressed in. And I won't click on correlated genes because that takes a while to load, but I'll tell you that it's looking across all our public data, and it's telling which genes are up or down regulated together. And we've also recently included interactions, protein-protein interactions, but I don't have any for this. So it looks like we are coming up to the point for questions and answers. So what I think I'll do is open it up. If you have any questions, we can talk about, I can try to help you with them. So preferably they say to do it through chat. So what I'll entertain your questions if you guys have any. And I know it was fast, but I'm happy to help. It's what I'm here for. I can show you one other thing. Seeing as I'm not being bombarded by questions, let me show you one other thing that you can do. If you have a FASTA file for a genome, what I showed you originally in the proteome comparison. So we stepped through using a reference genome and comparing it to a number of genomes, and that reference genome could be a public genome, a private genome, or a genome that was annotated outside of PATRIC. But for selected genome, it's got to be a PATRIC genome. I'm going to show you now what it looks like if you chose a genome that you had annotated outside of PATRIC. So I would choose, for this, I'm choosing the same folder, and TN PATRIC annotation. So the example I'm showing you is you can upload sequences into the database that you have that are not in the PATRIC website. So so to do that, and let me step back. Well, let me step back and show you, so I'll cancel it. So if you had something that was outside PATRIC, you'd have to get it into your PATRIC workspace from your computer. To do that, click on the folder, and that shows me all the folders and data I have in PATRIC, but I want to upload something new. So when I click on the upload icon, it takes me to a window where I can look for a particular file, and it's going to look for feature protein FASTA files. So if I go there, I have one here that I can upload. And so I say start the upload. And then you can see that that file is now the feature file that I'll be using as it was an annotation that wasn't from PATRIC. And now I can compare that to any genome that I'm interested in looking at in PATRIC. So I can make a quick comparison between those two versus, and then once I've added it to the box, I can submit it, and that job is off. So basically, today we've shown you, or I've shown you, different ways that you can add data to PATRIC and analyze it using this bidirectional blast hit. Although it was pretty quick, I showed you what it looks like in PATRIC, how it looks like in the workspace, how you can go from the visualization to a page in PATRIC, or from the visualization, or you can download the Excel file and examine all the detail, all the blast hits that are associated with each gene, and manipulate the Excel file to help you do that. And this is a great way to look for unique regions in different genomes. Let me point out, I pointed it out earlier in when I was talking. If you make a comparison, what we're seeing here in this particular example is based on, everything is based on this particular genome. So when I see things like this, that this genome is missing, that this genome has, I'm not seeing things that this particular genome, this 8T70 genome has, that the leopard TN genome lacks. What you would need to do is relaunch the tool using this genome as your reference genome with the same comparisons. I always do this. I take all the genomes that I'm interested in, and each one is the reference genome at some point, because I'm interested in finding all the areas that are unique within the entire pangenome of all of these organisms.